McLean. I'm going to be telling you today about some of the work I did for my PhD in Susan Albert's lab at Duke University. So one of the things that I'm most interested in is affiliative social behavior. Imagine a therapist could tell me why I'm interested in that, but as an evolutionary biologist, I'm interested in that because of the implications that it might have for how really complicated phenotypes like cooperation and altruism might evolve. So the affiliative social behavior that I looked at in my PhD is grooming behavior in primates. So here's two female baboons grooming each other. You're probably familiar with this idea, but grooming is more important than just removing ticks or other bugs. In primates, grooming is super important for social bonding. Even individuals in captivity where there's very little parasites spend most of their time grooming each other. We also know that grooming is really correlated with survival. So individuals who spend more time giving and receiving grooming are shown here in this blue line. See that? They survive better than individuals who groom less frequently, shown in red. Individuals who spend more time grooming also have higher infant survival. So here's our individuals who spend a lot of time grooming. Their infants survive better than those that spend less time grooming. This is true not just in baboons, but in all sorts of systems. Individuals who are more socially connected by a variety of metrics live longer lives. This is also true in humans. So we might, of course, expect sociality to evolve, for there to be a selective pressure. Individuals who are more social survive better, but we can't expect that unless this phenotype is explained by genetic variance in a population, right? This has to be reliably transmitted from parents to offspring. The variance that we see in sociality must be underlined by genetic variance in the population for this phenotype to evolve. So that was the first thing I wanted to test in our population. Is variance in grooming behavior explained by genetic differences between individuals? What population am I talking about? I worked on these baboons that live in Amboseli National Park in Kenya. This is a long-term field study run by my PhD advisor and this other group of fantastic women. Most of the data that I used for my study was collected not by me, by a fantastic group of field researchers and US-based scientists who've been monitoring these baboons almost daily for almost 50 years, collecting all sorts of behavioral and genetic and morphological and demographic data in a really non-invasive way, so the baboons are well habituated to human presence. They behave just as if we weren't there. We collect most of our genetic data non-invasively through fecal samples, which is very glamorous work. And I really can't stress enough how much of my work depends on the work of people who have come before me. There's no way I could have tackled this sort of analysis without lots and lots of dedicated people, dedicated to collecting data and storing it carefully. And it's a lot of work. Okay, but I did, luckily, have access to this data. And one of the great features of our population is we have a really extensive pedigree. We know who was related to who through microsatellite data and behavioral observations. And I could use a statistical method, commonly called the animal model, to leverage this pedigree to detect genetic variance for traits in our population. So I don't want to get really in the weeds about statistics here, but every result I'm going to show you depends on this, and so I'd like to make sure we all kind of understand it. So the animal model is just a mixed effects linear model, where in one of your random effects, you can incorporate the pedigree structure of your population. So you can think of random effects as describing sources of variance in your population. We call it a mixed effects model because you have random effects and fixed effects. The fixed effects you can think of as variables that might influence individual phenotype in an incremental sort of way. I'm not going to talk about the fixed effects that I included in this analysis. For those of you that care, I did calculate my heritability estimates with and without fixed effects and found very little difference. Okay, so that's sort of what the animal model is. How do you use it to calculate heritability? Well, you basically just construct a series of models with increasingly complex random effects structures, and then you test those models to see if the ones that include your pedigree structure are better by likelihood ratio test or information criterion or something than models that don't include your pedigree structure in the random effect. If so, then you can ask, well, what proportion of the variance in this population is explained by including that pedigree structure information. Okay, so this is what I did, and let me show you what I found. So far I've been talking about grooming as sort of a monolithic phenotype. I really divided it into two measures. I measured both the grooming an individual gave and the grooming that she received. So that's visualized here. So this is a baboon named Omo. These are all her social partners in any year of her life, in a given year of her life. My grooming given metric would be basically the sum of the widths of these green arrows for that year. My grooming received metric would be the sum of the width of these orange arrows for that year of her life. Okay, so that's the phenotypes I was looking at. What were the sources of variance in those phenotypes? So I constructed that series of increasingly complex models. My first model just included sources of error, like residual variance and individual ID. So this tells me here that this phenotype is pretty repeatable from year to year. Who you are explains some of the variance in this phenotype. My next model, I added a random effect of maternal ID. It suggests that the mothers that make up this population explain some of the variance in this phenotype. My most complex model included the pedigree structure, and you can see this pedigree structure is explaining some of the variance in our model, or in our phenotype. Did the same thing for grooming received, constructed that same set of models. In both cases, the model that included the pedigree structure was the best model, and we could detect that these phenotypes were indeed heritable, which is kind of exciting, especially for a wild population. And these are heritabilities that aren't super high, but on par with other fitness-related traits in the wild. Okay, so that's part of what I did. I want to spend a little bit of time talking about indirect genetic effects, because these are really perhaps especially important for social behaviors. So what are indirect genetic effects? You can think of an indirect genetic effect as the effect of someone else's genotype on your own phenotype, or reciprocally, the effect of your genotype on someone else's phenotype, right? So I might normally think, oh, I do what I do because of my genes and my environment, but my environment includes genes from other individuals, right? So those other genes can influence my behavior. Why might we as evolutionary biologists care about indirect genetic effects? This is really sort of fascinating to me, or it was when I first heard about it. Became less fascinating over time, but that's okay. When the direct and indirect genetic effects on a given phenotype are linked, how a population evolves phenotypically might be different than how it evolves genetically. So I want to try to walk you through an example of this. You can read about it if I do a bad job of explaining it. But imagine a population of mice, some variation genetically for how aggressive a mouse is in this population, right? Here's a mouse that has a high aggression allele. He's carrying that allele. The direct genetic effect of that allele, or genotype probably is better to say, is that this mouse exhibits increased aggression. But of course, you are aggressive towards another individual, right? So partner individual shows up. This mouse, who knows what its genotype says about how aggressive it should be, but just by encountering a mouse that's carrying the high aggression allele, this mouse's behavior could change, right? In this case, what Wilson et al. Found was that this mouse would become more aggressive. So there's an indirect genetic effect of the genotype of this focal mouse influences the phenotype of this partner mouse, right? So you can imagine then maybe there's selection on aggression, maybe more aggressive alleles increase in the population in the next generation, but then your population is going to become actually kind of increasingly aggressive above and beyond what you might expect just by genetic evolution, because even the mice that don't have the aggressive alleles are in an aggressive social environment. They might behave more aggressively than they would if they were in a peaceful social environment, right? So you have some genetic increase of aggressive allele frequency, but a really strong phenotypic increase in aggression in your population. Okay, so I wanted to look for indirect genetic effects on grooming. How did I do this? Basically, I altered my grooming phenotype a little bit. I kind of zoomed in on one of these green grooming given arrows and looked at it at the level of the dyad. So here's Omo, the amount of grooming she gave to this partner, happens to be named Ostrich. I assumed or modeled this grooming given phenotype in two components then. I thought maybe Omo has a genetic tendency to give grooming, like to push grooming on to Ostrich, but maybe Ostrich also has a genetic tendency to pull grooming out of her social partner, right? A tendency to elicit grooming. So it's an example of an indirect genetic effect. So my first test was, do indirect genetic effects even exist in our population? Can we detect them? So I did the same sort of thing. I made a series of models. Some included direct genetic effects, some included indirect genetic effects, included both. I'm just showing you the best model here. Was outlined are the direct genetic effects on dyadic grooming given and the indirect genetic effects on dyadic grooming given. They're small, smaller than the heritabilities I showed you previously, but they exist. This is the best model. Sort of exciting because with a number of caveats, these are some of the first, perhaps the first indirect genetic effects on affiliative social behavior in a wild population that we've detected. And what's really interesting to me is that we can tweak the animal model further and look for not just genetic variance between traits, but genetic variance of traits, but genetic covariance between traits. And we find good evidence that the direct genetic effect on grooming given is correlated with the indirect genetic effect on grooming given. So what this implies is that individuals who are genetically predisposed to give grooming are also genetically disposed to elicit grooming from their social partners. We might see accelerated phenotypic evolution of affiliative social behaviors. And what I'm really interested in exploring further is the idea that maybe this sort of linkage could make it more difficult for cheaters to invade a cooperative system if you're inclined to give and receive help genetically. And with that, I want to thank all the people who helped with this work, who funded me, and I don't know if I have time for questions. Yeah.
Do you know what the average relatedness is between the giver and the receiver?
So baboons do groom their family a lot more than they groom other individuals. This could have important implications for our analysis. I tried to control for that a lot in the fixed effects that I included. So given the fact that you have this much family, and in the dyadic analysis, I did control for that with exact pairwise relatedness. Yeah. Yeah.
Do you have enough power, do you think, to look at grooming given and grooming received, sort of the connection between them?
Oh, I did do that. They're very strongly correlated. Yeah. Thank you.